When people think of biotechnology in healthcare, they're often thinking of diagnostics or therapeutics, which is really the patient-facing side of healthcare. But there's a whole side of healthcare that's not obvious, but that's really critical for societies to be stable and resilient. And that's research capacity and also discovery capacity. I'm Keith Pardee, Associate Professor at the Leslie Dan Faculty of Pharmacy at the University of Toronto. Our lab is focused on improving access to biotechnology for human health. And we do this by using cell-free synthetic biology to produce proteins so we can bring biomanufacturing to the point of need. So in life science research, you really need bioreagents. They are the fuel or the lifeblood of life science research. And typically, when you want to make protein today, just like you would with brewing a beer, there's large microbial vats that produce lots of cells that express the protein of interest, and then you extract the protein from those cells and purify it. Right now, if you live in urban high-resource environment, these are things that are available within days. But in large parts of the world, low and middle-income countries, it can take months for these to arrive. They usually cost 2 or 3 times what they do in Canada. And because the cold chain can break down along the way, they're often of poor quality.
When I was a PhD student in Brazil, for most part of the time, I was buying my reagents, waiting for those reagents to do my experiments. And it was very frustrating. Later on, I realized that's it's a daily question and problem for all the scientists that work in the Global South. The people, they have the talent, they have the ideas, they have the knowledge, but they don't have the tools.
In cell-free systems, what we do is we take that molecular machinery out of the cells so that you can get rid of all of that infrastructure that you would need for producing protein conventional methods with cells. And you just freeze dry it down into a pellet, and you can ship it like ramen noodles at room temperature. You just add water, and when you get to wherever you need to be, you just add water, and within a minute, you're starting to make proteins in these systems.
The primary goal of this project was demonstrate that we can produce the proteins on site. And actually, we did at 10 sites across 3 continents, including in the top of a mountain in Yukon.
Where we see real sort of the next steps for this project are to automate cell-free biomanufacturing. You can think of it just like an espresso machine. You know, with the push of a button, you're not making coffee, but you're making an antibody, or you're making an enzyme. And so these would become basically point-of-need production where they could make lab reagents, diagnostic reagents, or even in the long term, therapeutics for patients.